Welcome to our talk on Porifera and Placozoa. This is the first category of two animals that we're going to talk about. Porifera, or what we also call sponges. I'll use the term interchangeably. In this lesson, we will explore the phylum Porifera and phylum Placozoa and look at the organisms that make up their group. We'll also discuss here the origins and their key features and discuss why they're important to the oceans. Before we start, allow me to introduce myself. I am Enrico Gutierrez Media, your presenter. In today's topic, let us be guided by the following objectives. First, trace the origin of animals. Second, discuss phylum Porifera sponges and phylum Placozoa. Lastly, describe the phylogeny and adaptive diversification of sponges and Placozoa. To start with, most major animal groups around today originated in the Precambrian and Cambrian era. Many of the Cambrian species are extinct, but their body plants remain species alive today. The sponges first appear in the fossils record in the Precambrian, and the oldest known fossils are about 600 million years ago. Although a well-preserved fossils of an Idicarian sponge was described in 2015 from the Shuanto Formation of South China. The sponges and placozoans represent the simplest of all animals. Sponges appear to represent an early stage of multicellularity in the animal clade. Although they have specialized cells for particular functions, they lack true tissues in which specialized cells are organized into functional groups. Sponges are similar to what might have been the ancestor of animals such as colonial and flagellated protists. The Placozoa members have the smallest nuclear genome and the largest mitochondrial genome of any known animal. Their circular mitochondrial genome shares some features with mitochondrial genomes from animal outgroups, including chytrid fungi and quanophyllate, that also has derived animal features. So everyone probably knows who SpongeBob SquarePants is, right? Well, did you know that he could also be known as Porifera Bob SquarePants? Well, the term Porifera is actually the scientific name given to the group of organisms known as sponges. But unlike SpongeBob, living sponges are neither square in shape. They actually important and diverse phylum within the animal kingdom. That's right, sponges are actually living animals. We are going to learn a bit about them here. Evolution of eukaryotic cell was followed by diversification into many lineage. Modern descendants of this lineage includes unicellular taxa, as well as colonial and multicellular plants, animals, and fungi. Animals traditionally were divided into single-celled animals or protozoans and multicellular animals or metazoans. Because protozoans belong outside the animal kingdom, metazoan is now synonymous with animal. The sister group to the animal is the iguanoflagellates, according to several phylogenies using molecular characters. Iguanoflagellates are solitary or colonial aquatic eukaryotes, with each cell carrying a flagellum surrounded by a collar of microvilli. Beating of the flagellum draws water into the collar where microvilli collect tiny particles, typically bacteria. Many iguanoflagellates are sessile and attached to hard surfaces. Iguanoflagellate cells are strongly resemble sponge-feeding cells called iguanocytes. Now, was a sponge iguanocyte inherited from a common ancestor with iguanoflagellates? Arguments against this hypothesis includes the observation that iguanocytes occur only in adult sponges and are not part of the early developmental sequence. Instead, flagellated cells without collars develop into guanocytes after larval metamorphosis. Collar cells also occur in certain corals and some echinoderms, so if they were part of the earliest animal lineage, this morphology has been lost or suppressed in most taxa. Now, despite these objections, there is another clear link between guanoflagellates and animals. The protein used by colonial guanoflagellates for cell communication and adhesions are homologous to those that animals use in cell-to-cell signaling. In order to form multicellular body, cells must adhere to each other in a stable fashion. The proteins used in cell adhesions and cell signaling in sponges are homologous to those in other animals. In fact, many of these are present in guanoflagellates, evolving before the last common ancestor of all animals. The animal epithelium is a unique structure where cells adhere to each other in a single layer attached to a basement membrane that contains collagen type 4 adherence junctions in which coherent molecules function as adhesives, which connects in an epithelium to the basement membrane. But a true epithelium also requires the cells to link to each other using particular junctions called desmosomes. Because the homocellulomorphs do not have these later junctions, but to have the former adhesive junctions. Now, some researchers consider spinacoderm is a true tissue. However, in a true tissue, the cells connect to each other via proteins called adherents, in particular junctions called desmosomes. There are no desmosomes in homocellomorph pinacoderm, so it may be correct to call pinacoderm an incipient tissue, not a true tissue. To identify the more complex regulatory pathways in how they evolve over time, and this is because even in sponges, they exhibit anterior posterior axis in their larvae, even though they're simple and don't have a whole bunch of other axes, they do exhibit anterior posterior axis. And then if we're looking at the sponge larva, and then we kind of look at their organisms over time, more and more complex over organisms, we might be able to get some idea of how these complex regulatory pathways began. There is a debate as to whether sponges have true tissue epithelia that are elaborated in other animals. It is possible that sessile lifestyle of sponges favored a deceptively simple body in most species and that a closer look at sponges will reveal more some features typical of animals. Now, in phylogenetic studies using sequence data from large subunit of ribosomal RNA, small subunit of RNA, and protein kinase C, which indicates that sponges with calcareous spicules in the calcipongiae belongs to a separate clade from those with spicules made of silica in classes Thymospongiae and Hexatinellida. Two potential placements emerge for calcareous sponges. One in calcareous sponges are the sister taxon to a clade of siliceous sponges, and in another, the phylum Porifera is a paraphyletic because the calcareous sponges are more closely related to the animal taxa than they are to siliceous sponges. Now, the name phylum Porifera simply means spore bearing. Sponges are the sister taxa of all animals, so they kind of the most base or the plain animal, the first kind of branching off when they were looking at the taxonomy of animals, and they represent a similar stage of complexity. So they're composed of different individual cells. They are associated with each other through extracellular matrix, but their individual cells perform independent. They have very simple morphology, so they are not complicated animals to follow. They have various shapes. Most are asymmetrical, but some can be radial. And if you look at many pictures of sponges on the internet, you'll see the diversity of sponges. Sponges don't have defined symmetry. All sponges are sessile, but they do have a free swimming embryo and they have no mouth nor digestive tract. So how do they feed if that's so? Well, through suspension feeding. Some capture prey, but the vast majority of them undergo suspension feeding, like I mentioned. They can vary in shape and they can also vary in sizes and many colors. You can have sponge that more like a mat of moss, but actually a sponge. Other sponge have this large tubular shape, like you see in the picture here. Some can also like a fan. They have all types of morphology, and some can be tall, like meters of tall. Some can be thin and small, and they can have various colors, right? Right. So there is a lot of different diversity of sea sponges under the oceans out there. Most of them are marine dwelling, but they can live in fresh water environments and also in brackish water. Porifera or sponges have some skeletal framework structure. It can be fibrous or rigid, but they don't have necessarily true skeleton on it, but rather they have this some sort of fibers that are called spicules, and they can be made of silica or they can be made of calcium carbonate. This provide them some sort of their support in their structure as sponges. Otherwise, they would probably be like jelly. The fibrous part of the skeleton comes from the collagen protein fibrils in the intercellular matrix. Collagen differs in chemical composition and form. One form of collagen is called spongen. The classification of sponges is based on their spicules, form, and chemical composition. Genetic analysis also indicates that calcareous spicule sponges and silicate spicule sponges have their own clade. Sponges harbor microalgae and cyanobacteria on the body surface and deep inside their body. Sponges feed primarily by collecting suspended particles from water pump through internal canal systems. Water enters canals through a multitude of tiny incurrent pores in the outer layers of cells, known as pinacodia. Incurrent pores called dermal ossia have an average diameter of 50 micrometer. You may be surprised to discover that a large sponge can filter up to 20,000 liters of water each day and they non-selectively consume food particles. Now, there are three types of sponge structure: acidoids, synconoids, and leuconoids. The degree of complexity from simple asconoid to complex leuconoid type has involved mainly the water canal and skeletal systems accompanied by outfolding and branching of the collar cell layer. Asconoid sponges have the simplest organizations among the three. The sponge draws water inside through the microscopic dermal pores by the beating of large numbers of flagella on the choanocyte. These choanocytes line in the internal cavity called spongicle. In synconoids, sponges look somewhat like a larger asconoids. They have a tubular body and single osculum, but the body wall, which is really the spongical lining, is thicker and more complex than that of the asconoids. The myconoid type is considered a major plant for sponges for it permits greater size and more efficient water circulation. On the other hand, leuconoid sponge body comprises an enormous number of these tiny chambers. Clusters of flagellated chambers are filled from in-current canals and discharged water into ex-current canals that eventually led to an osculum. Now, a sponge pumps a remarkable amount of water, about 2,000 liters of seawater. However, because water passes into flagellated chambers with a greater cross-sectional area than those of the entry canals, water flow through the chambers slowly. Such a flow rate allows the ample opportunity for food captured by the choanocytes in the flagellated chambers where the food collection occurs. After the food is removed, the used water is pulled to form an exit stream so that water exits from many local sites on the sponge. Most sponges are of the leuconoid type. Leuconoid bodies account for most species within a class of Calcipongiae and are the most common types in other classes. Now, there are four major sponge shells, which are pinacocytes, porocytes, amoebocytes, and choanocytes. The small section through a sponge wall showing four types of sponge shells on the picture. Pinacocytes are protective and contractile. Choanocytes create water currents and engulf food particles. Archaeocytes have a variety of functions. Colonocytes are which secretes collagen. Sponges lack structured tissue, so they depend on specialized cells like choanocytes, porocytes, amoebocytes, and pinacocytes in order to perform their specialized functions. Now, porocytes regulate the amount of water that reaches the spongicle pores, while choanocytes, which are flagellated cells, help the sponge transfer water through the sponge, allowing it to trap and digest food particles. Now, in sponges, pinacocytes are a form of epithelium. They involve in cell adhesion, signaling phagocytosis, polarity, and movement. Pinacocytes are packed with mesohyl, a gel-like material that helps the sponge retain its shape and its structure. Amoebocytes, on the other hand, have various functions, including delivering nutrients from choanocytes to the other cells, forming eggs for sexual reproduction, delivering phagocytated sperm from choanocytes to eggs, and transforming into different cell types. Choanocytes are sponge cells with a central flagellum or cilium surrounded by a collar of microvilli bound by a thin membrane that lines the interior of the asconoid, synconoid, leuconoid body forms. And sponges have also a sexual and sexual reproduction. A sexual reproduction includes budding, regeneration, and genios. It occurs by budding or by fragmentation. Now, the buds may remain attached to the parents or separate from it, and each bud develops into a new individual. The internal buds, which are formed by the freshwater sponges, are called genios. Sponges can also have tremendous ability to repair injuries and restore lost parts, and in a process called regeneration. In a sexual reproduction, uh, most sexually reproducing poriferans are hermaphroditic, meaning they develop eggs and also sperm at different times. Sperm are released into the water column daily. The sperm are produced concentrated and released through the excurrent openings of an dense mass, give to the sponges an appearance of smoking. Now we're going to discuss the taxonomy of phylum Porifera. First, the class of Calcispongiae, or the Calcispongiae are calcareous sponges, so they're called that because their spicules are composed of calcium carbonate, which are often form a fringe around the osculum. Spicules are straight needle shaped or have three or four rays. These sponges tend to be small, like 10 centimeter or less in height, and tubular or vase in shape. They may have all three types of canal system, asconoid, synconoid, and leuconoids in structure. They are all marine species, although they may are drab in color. Some are bright yellow, red, green, or lavender. Examples is Clathrina, which is a small with intertwined tubes. Other is the class of Hyalospongia or Hexatinellida, the glass sponges form. They are nearly all deep sea forms that are collected by dredging. Most are radially symmetrical, with vase or funnel-shaped bodies usually attached by stalks or root spicules to a substratum. Their distinguishing features include skeleton of six-way siliceous spicules that are commonly bound together into a network-forming glass-like structure. Classic example of Hexatinellida is a Venus flower basket, the lattice-like network of spicules found in many glass sponges, of which it is an exquisite beauty. The other one is Plasmodium spongiae, which make up this group contains 95% of living sponges species, including most large sponges. Their spicules are siliceous, but they are not six-ray. Spicules may be bound together by spongin, or they may be absent. So-called bat sponges, spongia, and hippospongia belong to the group called known as horny sponges, which have been spongy skeletons and lack siliceous spicules inside. All members of this class are leuconoid, and all are marine, except for members for the freshwater family Spongilidae. Marines, the most spongy are quite varied and may be striking in color and also shape. Some are encrusting, some are tall and finger-like, some are low and spreading, some bore into shells, and some are shaped like fans, basis crustaceans, or balls. Freshwater sponges are widely distributed in well-oxygenated ponds and streams where they can encrust plant stems, and old species submerged wood. They may be resembled bit in wrinkled scum, bit filled with pores in the brownish or greenish in color. Now, the last class for the Porifera are the Homoscalomorpha, or the Homoscalomorphs are marine sponges that occur in a range of colors but thrive in cryptic habitats, so they are often overlooked. They are more common in nearshore habitats, but they do occur in deep water. Sponges in this class were formerly placed in class Hemospongiae, but have been separated because they possess unique features such as pinacoderm layer with a true basement membrane. The cells of this class is layer, which also differ from those in cells of other sponges no class, because they connect not only to each other but also to the extracellular matrix with special adherence cell junctions. That's the end of the phylum Porifera, and moving forward to the phylum Placozoa. The phylum Placozoa are extremely simple animals for which there is a little understood due to lack of observation in their natural environment. In nature, no one knows what substrate they live on and what they consume. It's also unclear if they reproduce sexually, as do most species. No, they were found on the glass walls of an aquarium in the European laboratory in the late 1880s. Since then, much of what we know about their biology has come from researching cultures that have been kept alive in laboratories all over the world. Placozoans have yet to be discovered as fossils. Considering their small sizes and squishy appearance, it is unclear what Placozoans eat in the wild. They could eat variety of species. The phylum Placozoa was proposed in 1971 by K.J. Grell to contain a single species, a Trichoplax adherens, a tiny marine form of about 2 to 3 millimeter. Their body is a plate-like and has no symmetry at all, no organs, no muscular or nervous system. It also lacks both basal lamina beneath the epidermis and extracellular matrix, two features that were considered animal hallmarks. However, the genome contains many genes that encode predicted extracellular matrix proteins. Placozoa has a sandwich-like body plan with a lower epithelium and an upper epithelium. A loose layer of so-called fiber cells is encased between these two layers. Placozoa does not seem to have any symmetry or body axis. The body placozoan is composed of a dorsal epithelium of a cover cells and shiny spheres and a thick ventral epithelium containing monociliated cells and non-ciliated gland cells. The space between the epithelium contains multinucleate fibrous contractile cells. There are four types of cells distinguished morphologically in the Placozoa, but gene expression studies suggest the presence of a fifth type. Placozoa feed on waste material, protozoa, and algae. Placozoans glide over their food, secrete digestive enzymes on it, and then often absorb the products. In the lab, they feed on organic matter and small algae. The life cycle of Placozoan is not completely known. In reproduction, they divide asexually and produce warmer stages by budding. Although sexual reproduction has not been observed, eggs occur in the laboratory animals. Genetic studies of Placozoans from around the world show that 8 distinct lineage equivalent to species exist. Although they cannot be distinguished morphologically, sexual reproduction has been inferred from molecular evidence of genetic diversity within a clade. Grells considers trichoplax dipoblastic with dorsal epithelium represents ectoderm and ventral epithelium representing endoderm because of its nutritive function. Gene expression studies support these homologies. The origin of the middle fibrous layer is currently under study. At present, we depict branching of Placozoans, Cnidarians, and Ctenomorphorans as a polytomy. For the research update, the research article entitled A Collection of Hexaptenalids Porifera from the Deep South Atlantic and North Pacific, New Genus, New Species, and New Records by Dr. Christiana, Dr. Allen, and Dr. Eduardo. The research article was published on July 9, 2020. This article describes the four hexatenolid sponges, namely Poliopogon amado, Eupelectilia centipoli, Bolosoma perezi, and Adhena magnifica. These represent new findings for the South Atlantic deep sea fauna, including the first record of Bolosoma for this ocean. Adhena magnifica, on the other hand, was collected by NOAA oceanographic expeditions in the North Pacific. In the introduction, in deep sea environments, corals and sponges form ecological important structures that provide habitats for organisms. Hexatenolids or glass sponges are siliceous marine sponges that occur in both hard and soft sediments. These sponges are diverse and abundant in deep sea environments. Despite their ecological importance as ecosystem, engineers providing framework habitat for many taxa, hexatenolids most likely have their diversity severely underestimated. The entire South Atlantic has had only 30 species reported to date. The majority are 28 from southwestern Atlantic and most occurring on the continental slope. To increase knowledge of hexatenal biodiversity, we describe here new species and new records based on dredging, rogue operations, and manned submersibles conducted in two sectors of international seabeds. In their methodology, data were presented here came from Pro-IRG data and deep water exploration of Marianas and Deep Discover Rogue Oceanographic Expedition. The first two to the Rio Grande Rise and Sao Paulo Ridge in 2011 and 2013, and the last one was on 2016 on the Marianas Trans-Pacific Ocean. These specimens are deposited partly in the Porifera collection of Museo and partly in the Smithsonian National Museum of Natural History collection. The material was studied described the following standard procedures outlined by Riswig and Stone, with the difference that isolated spicules was not captured in nitrocellulose filters, but were pipetted directly onto glass slides or metal stubs. In the results and discussion, the Foliopogon amado, it was a lopophytus sponge, semi-funnel to fan-shaped body, following a leaf rolled up in a semi-funnel with concave gastral and convex external surface. Like here in the photo, the specimen is 173 millimeter and 67 millimeter in diameter, adjacent to basilia. It was compressible and rough. Basilia composed of broadstuff of anchoring spicules. They are in brown color in situ and after preservation in ethanol. The other one is holotype of Eupalactilia centopoli, the only known Eupalactilia without sieve plate, at a principal skeleton composed by long, smooth stauractins, guanosomal hexactins, and pentactins. The sponge consists of delicate thin walled tube bearing a conspicuous surface articulations such as in the photo with a spurs of oscula up to 4 millimeter in diameter and basilia in a single tuft. It was a holotype, 137 millimeter long and 32 millimeter in maximum diameter. The other one is Bolosoma perezi, the only Bolosoma with discohexasteres and codonhexasteres microscillers. The Bolosoma comprises 8 species, up to now recorded only from the Pacific Ocean. The new species presents a unique set of spicules, indicating its distinctiveness from the remaining spicules in the genus. None of it is congeners, present the combination of delicate codonhexasteres and ball-like discohexasteres, which confers Perezi obvious status as a new species. This is the first record of Bolosoma for the Atlantic Ocean. The last one is Advena magnifica, or the ET sponge. Advena magnifica is the only representative of Bolosomia with microscleres and pedunculate sponge characterized by a mushroom like. Considering all genera of Bolosomia, Advena appears most similar to Neocaledoniale, uh, Tabanik, and Levy. The only other Bolosomine with calicocombs and graphiocombs. Thus, they propose a new monospecific genus within Bolosomia to include Advena magnifica. For their conclusion and recommendation, a new species of Bolosoma is described in raising to 9 the number of known species in this genus and expanding the genus known distribution to the Atlantic Ocean. A better assessment of deep sea sponge biodiversity in the Atlantic and the Pacific will only be possible with additional efforts, both enabling new expedition but also granting support to the necessary work on proper identification and collections already available. A considerable number of deep sea sponge specimens is deposited in scientific collections around the world, calling for taxonomists to work on them and furnish not only inventory data but also reveal surprising animals whose integrative study can help us build a sharper Porifera tree of life. So that's all for the phylum Porifera and phylum Placozoa, and I would like to end this with this quotes from SpongeBob. Stated that no one can change a person, but someone can be a reason for that person to change. For that, thank you for listening and continue to inspire our students in these trying times.